Namaskar. Today we will study the difference between the sense strand and antisense strand. So this sense strand, antisense strand, that comes under your deoxyribonucleic acid, and we know the DNA is a double strand helical structure where adenine of one strand forms pairing with thymine of the other strand by double bond, and cytosine of one strand forms triple bond with guanine of the other strand. And one strand is in the direction of 5' to 3' and the other strand is in the direction of 3' to 5'. And what is this 5' to 3' direction? If you see the structure of the sugar, in DNA we have the deoxyribose sugar, and here this is the structure 1', 2', 3', 4', 5'. So here you can see the structure of one nucleotide of DNA. Here the base is adenine, the sugar is deoxypentose, and it is linked to this 5' position, to this 5' carbon, this phosphate is linked. So this phosphate can combine with the 3' position of another sugar, and here to this 3' position one phosphate can attach, which this phosphate will be linked to the 5' position of another sugar. So in this way, suppose this is here to this phosphate, if there is linkage of this 5' position of another this will be linked to the 3' position of another sugar. So that means this one, suppose this sugar comes here, so this O means this is linked to this one, this third, and similarly this third one will be linked to the fifth carbon of another sugar. So in this way we can have one 5' to 3' direction, and we can have another one from 3' to 5' direction, and the strand with from 5' to 3' direction is called the sense strand or coding strand or plus strand, and the strand from 3' to 5' position is called the antisense strand or template strand or non-coding strand or the minor strand. And why we call it as the sense strand? Because this DNA will encode the messenger RNA, and that messenger RNA will have the same base sequence as that of the DNA, this strand, sense strand. Only the thing is, instead of thymine, the messenger RNA will have uracil. So how this antisense strand will act? This antisense strand will act as a template, so that the complementary of this one, which is same as that of the sense strand, will be there, but instead of thymine there will be uracil. So suppose this is the antisense strand, so here T, the complementary is A, so messenger RNA will have A. Again it has A, the complementary is T in case of DNA, but it will be U in case of RNA. So let us see that. So this is say 5' to 3' direction, this is the sense strand, and 3' to 5' direction, this is the antisense strand. So by the action of DNA gyrase, both the strands are separated, the sense strand and the antisense strand. Now this antisense strand, you can see complementary is T to A to U, C to G, T to A, like this, and this is messenger RNA. And if we will compare this messenger RNA base sequence with that of the sense strand base sequence, it will be almost same. Only difference is in the sense strand we have thymine, and in mRNA it is uracil. So if we study the difference between the sense strand and the antisense strand, sense strand is also known as plus strand, antisense strand is known as minor strand, and sense strand is mRNA like strand, only the difference is thymine is replaced by uracil, and it is the source from which your protein synthesis occurs. So it is also called as informational strand, and since the antisense strand acts as a template for the mRNA synthesis. So it is called template strand, antisense strand is called template strand. So we can say sense strand as the non-template strand. And again, similarly, we can say sense strand as the coding strand because it has the code for the protein to be synthesized, and antisense strand hence called as non-coding strand. It is only acting as a template. The base sequence is same as messenger RNA except uracil, as we have discussed, because that's why we call it as mRNA like strand, and in antisense strand acts as a base for mRNA transcription. So the antisense strand by the action of DNA dependent RNA polymerase, the complementary bases are formed that forms the messenger RNA. So it acts as a base for the synthesis of mRNA, whereas the codes are there which is almost similar to that of messenger RNA. Only difference is thymine is replaced by uracil. So that's why we call the sense strand as the coding strand. So the sense strand contains codons, the antisense strand contains anticodons. As we can see here, now let us see this messenger RNA then translated by transfer RNA. So the codon, the first three say AUG is the codon. The anticodon is with the transfer RNA. At one side there is amino acid and the other end there is the anticodon. This anticodon will recognize the codon. So A for U, U for A, G for C, UAC. And if you see that with the compare it with the antisense strand, here it is TAC because it is DNA. And if you replace this T by uracil, then it will be UAC with the transfer RNA. So it has the anticodons in case of antisense strand, and the sense strand direction is 5' to 3', antisense strand 3' to 5' direction. And since the sense strand has no role in your mRNA synthesis, so no hydrogen bonds between sense strand and synthesizing messenger RNA, whereas temporary hydrogen bonding is there between the nucleotides of the antisense strand and the synthesizing messenger RNA. So this is all about the difference between the sense strand and antisense strand. And if we summarize, so the strand from 5' to 3' direction is the sense strand, and the strand from 3' to 5' direction is the antisense strand. The sense strand has the code, the codons which are transferred to the messenger RNA. So messenger RNA will have the same base sequence as that of the sense strand, only the difference is T will be replaced by U, and the antisense strand acts as a template for that messenger RNA synthesis. So the complementary bases of the antisense strand form the messenger RNA. So that's why the messenger RNA has the same base sequence as that of the sense strand or coding strand. Thank you.